Welcome, everyone. Thank you very much, Alex and Dieter, for agreeing to give a Build a Cell seminar. And it's all yours.
Yeah, I mean, this is Alexander Floroni and Dieter Braun. We are Origin of Life group in Munich, and some of our explorations go towards synthetic cells. So he'll do most of the talking, and if you want our CVs or what we did in the past, check the websites. So we come from the origin of life question, where you have an early Earth, and you think about how can these conditions lead to first cells. So it's not a synthetic cell approach, it's kind of the real cell approach, but of course we might have a different twist in the way that there's no pipettes, no labs, biochemistry lab on early Earth. So we tried to think how that could work about. So if you look for the timeline, just give you a little bit intro on the origin of life perspective. Proteins at some point emerged and likely have revolutionized how cells made metabolic networks and could steer all those funny, nice metabolisms, which of course imply that the proteins, since they're made out of ribosome and tRNA, you must have had some RNA before. So we follow that paradigm and think about nucleotides, especially the 2,3' cyclic we like a lot, to establish RNA evolution. So that's where we do our major business. And then you think about where do the first vesicles come about. And there I'll just switch to laser pointer. So there the thing is that we'd love to see these pores on early Earth to be the first hatcheries of life, and therefore will not put those vesicles early on. But of course, this only would make sense if those conditions back then, and especially non-equilibrium conditions, because all life needs non-equilibrium to run, could make the first vesicles. And that's basically what Alex will talk about, where we are in that realm and where you can make vesicles, but most of all, that you can get away without vesicles, and where we are there, but how that bridges together. So the hypothesis of early life, I've just pushed this through that if you have not heard it, we think these non-equilibrium conditions can accumulate molecules, especially those who can polymerize, because they will be then accumulated, making short strands which ligate, and that would self-purify into either right or left-handed strands. That would be the idea. Where later life has selected one of them. These ligation chain reactions would form replication networks, which would self-select for length and speed. Again, in these pores, we think it's a good setting because they offer not only accumulation, but better accumulation for more complex molecules. And you'll see how that actually feeds into the story, that they become more complex and actually form cells eventually. All that would be in pores. It would populate those pores on volcanic islands. Actually, a little bit alkaline pH is good for this kind of chemistry. But the interesting thing is that that's what we'll talk about, that these conditions still could host modern life. So it would be a continuous, and they form these cells. And that's the idea, to recreate the origin of life on that. So today we focus on how cells are filled and how they can bridge towards cell biology. And I give it over to Alex.
Thank you, Dieter. Hello, everyone. So I'm just going to continue the talk here. So as Dieter mentioned, we are starting from an early Earth scenario. And while we think that the molecules that made the first organisms were there, they were probably not, cell-like concentrations as we see today in modern cells. So we assume that in the bodies of water that we had on the early Earth, be it the ocean or volcanic ponds, we had our molecules, be it nucleotides or amino acids or early lipids, a very dilute state. So in this very dilute state, they would be kind of in equilibrium. There would be no energy input, and without that, they wouldn't be able to accumulate and have interactions and create all those prebiotic molecule networks that Dieter was talking about. there would be no confinement, there would be basically nothing. So you would need some form of energy input on the early Earth that would lead to this non-equilibrium setting that current life is also working in. And that would lead to confinement, it would lead to those networks, it would lead to localization, and it will lead to potentially creating living cells. So in this early Earth, you have a lot of energy inputs that you can talk about, but the most prevalent one and the most ubiquitous one would be heat. And heat is a nice thing because heat is present uh, in a lot of different places on Earth and also on other planets. So we were not limiting ourselves to Earth here, be it from the sun or from magma in like geothermal context. What we are also talking about, and this heat usually leads to some form of temperature gradient. We will find this kind of heat, as I mentioned, a lot in places which have volcanic activity, for example, at the bottom of the oceans, at the rifts, but also on volcanic islands where you can have, those volcanic ponds, as you can see in Iceland or in some other places in the Yellowstone Park, which are quite hot, and they can also have this temperature gradients. And what those places have in common, wherever there's volcanic activity, there's also porous rocks. And those porous rocks can host a variety of different pore lengths and thicknesses, and a very interspersed network of them. And we are talking in our project about two specific mechanisms. On the one hand, you have this completely water-filled pores here, where you have a temperature flux from, in this image, for example, from the left to the right. And this temperature flux will create two different flows inside of these pores. And those two, one of them is thermophoresis, the other one, of course, is convection, since we live on a planet, so there's gravity. And these two flows together will lead to a net accumulation of molecules, which you see here in this nice simulation, on the bottom of the chamber on the cold side. And this is specific to the molecules, of course. So you can have molecules that have more stronger accumulation, ones that have more weaker accumulation, but you can accumulate everything. You can accumulate DNA, you can accumulate RNA, proteins, whatever it may be. The other mechanism that I'm going to talk about a bit later is accumulation at air-water interfaces. So in these situations, you don't only have pores that are completely filled with water. Of course, since there's heat, you also have the gassing of your water. So you will have also gas bubbles. And at these, if these gas bubbles become stuck in those pores, they will also have this temperature gradient flow across them. And this will lead to another form of accumulation. As you can see here in this diagram, you will evaporate water on the hot side, you will condense it on the cold side. You will have a capillary flow that will push your molecules to the hot side, and that will lead to also accumulation. I'm going to talk about this in more detail a bit later. For now, I just wanted to give you kind of more of a idea of what the mechanisms are and where we are in our temperature, in our early Earth setting. So the way we do this in our lab is with those thermal chambers, as we call them. They're basically a sandwich of a lot of different materials, but the most important part of it is the middle here, which is this Teflon sheet where we can cut our pore. So we can cut it the way we want it. We can test different sizes, different thicknesses, different conditions, different geometries. And we sandwich this together, heat it from one side and cool it from the other side, and create basically our rock pore simulant. And we use those transparent plates here on both sides of the pore in order so we can follow the accumulation of the molecules if they're fluorescing. We can see where the intensity will be localized, and the increase in concentration. And here you can see some of the studies where we did this. And as you can see in some of them, you can already see like how this would look like in a microscope. We look at it kind of from the side. So we look through the pore together, with the heat flow. So we will see the accumulation on the front of the chamber if it's air water, and on the back side of the chamber if it's the thermogravitational trapping. And we do this in our kind of self-built microscope. It kind of works a bit like a 3D printer, so it has some positions that you can set, and it will know at that point I will take images, and you can set how many images you want to take. You can set how many times that you want to take it, different LED intensities. And also, as you can see in this experiment rig here, you can fit multiple traps, so you can test different conditions, different solutions, and also you can heat up the traps at different front temperatures. The back temperature is together, this one block, but the front temperature you can also change in between them. So you can test a lot of different conditions at the same time, and you can all record them with this microscope here. So what we want to do with this, we did a lot of experiments in our lab with single molecules. So we accumulated nucleotides, we accumulated amino acids, we also tested polymerization reactions and everything else. But we also wanted to see what the limit is of this mechanism. So how many molecules can we accumulate at the same time, and can those molecules, after they get accumulated, even interact with each other? Do they even work? So what we want to do is we took a transcription translation reaction, which is quite a complex reaction, even by any standards. It has over 100 components, which all have to be there in order for this reaction to work in any efficient, in any meaningful way. And if you dilute these components, as you can see here, once you dilute it to at least 3 times, you will basically kill the reaction. So the molecules will be exactly as we envision them on the early Earth. They will be in a dilute phase, they will be separated, they will be in equilibrium, there would be no form of reaction going on. And what we want to do is using this initial state, using the accumulation mechanism that we have, to put all those molecules together and start the reaction. And the marker that we use for this is superfolder GFP, which is the standard marker in biochemical reactions. It folds easily, and it has a very, very bright signal. So the first thing that we tried was with thermogravitational accumulation. And if you remember from before, this is where you have the heat flow through a water-filled pore, and you have this thermophoresis and convection. So you'll accumulate the molecules on the cold side on the bottom. And as you can see here in this diagram, this will kind of look like this, where you have a very concentrated cell-like environment where your reaction is going on. And going away from that part where the molecules are accumulated, you get less and less and less and less until you reach kind of the other end, which is kind of like the environmental phase. And this is the separation that you have in modern cells, right? You have the cell interior, you have the cell exterior, but they're separated by a solid membrane. Whereas here, it's kind of like a continuum. It's like a non-material kind of membrane. And you can see this is how it would look when you take the microscopy images, and you can track the intensity of your superfolder GFP production throughout the entire trap. And you can see here, even with non-diluted pure reaction, which should produce superfolder GFP everywhere in the trap, you still see a localization of the superfolder GFP at the bottom of the trap, indicating that at least superfolder GFP gets accumulated if you put a thermal gradient here. What we can do with this trap afterwards is we can freeze it. We can cut it into 3 pieces, for example, here, in order to get some amount of volume that would be around 11 microliters of volume per phase. And we can use this volumes for other assays in order to test for different components, not only superfolder GFP. We can also watch the rest of the proteins. We can also track DNA and ions, which I'm going to talk about a bit later. What I wanted to say before is one of the aspects that influences this accumulation, as you can imagine, is temperature. And we chose our temperature here for pure to work in, because pure works best at 37 degrees, since it comes from cells. And we wanted to stay in that range with our temperature gradient. So we didn't want it to do it too high on one side and too cold on the other side, because that would lead to either the reaction not working at all, or even to the denaturation of the proteins, which would also permanently kill the reaction. So we stayed kind of in around 42 degrees, 40 degrees on the hot side, 27 degrees on the cold side. And this would be in a pore of like around 170 microns, which we also, found out experimentally, but also through simulations. So when we put diluted pure, of course, we don't expect it to work in bulk. And you see, after we put it in the trap and you turn on the temperature gradient, you see that the accumulation will lead to superfolder GFP being at the bottom here. And you really see here nicely that you really have a temperature gradient following concentration gradient following this double exponential curve here with the most accumulated material and the most superfolder GFP production at the bottom. And we know that it's the temperature gradient that's really doing this, because if you put it in a trap where you don't apply any temperature gradient, for example, as we did in this experiments here, at the extreme temperature, so 27 and 40, and at the optimal temperature, 37, you won't see any superfolder GFP production, indicating that you really need, the accumulation for superfolder GFP production. Otherwise, you wouldn't see anything. And it's not only accumulation that is important, but also confinement. So once you have your gradient, of course, everything will accumulate, your superfolder GFP gets produced. But then if something happens to the gradient, or if the gradient subsides, or if something happens in the heat flow, of course, your molecules that you accumulated in your entire reaction will start to diffuse away. And as you can see here, due to how the pore is built and how the diffusion works, it takes a long time for the reaction to reach uniformity after it has been accumulated. It takes, for example, in the simulation at 27 degrees, it takes about 3 months for it to diffuse and homogenize. And this will only take longer if you have a longer pore. So in order for you to kind of get an idea of what I'm talking about, this is kind of what we see if we stack all the images over the entire height of the trap. And in order for me to fit the video, I had to put it horizontal, but you have to imagine that the right side is the bottom and the left side is the top. And this is an experiment, for example, where we didn't introduce any temperature gradient. And as you can see, or maybe not see, there is no superfolder GFP production inside. So it remains completely dark. But if we add a temperature gradient, and as you see here, it's around 13 degrees, you can see that at the bottom of the trap, you have superfolder GFP production, and it only, there's a gradient from the bottom to the top where you have less and less superfolder GFP, which is quite nice that we can actually see how this would look like. I mentioned before that we can, in order to check all the other components, we have to kind of extract the different parts of the trap. And we do that by freezing, as I mentioned. We freeze the whole chamber, and then we slowly thaw it. And while we thaw it, we extract the different parts, and we use those parts for other types of experiments. And in this, for this, we used two controls. So one control would be the one that I showed before, where we don't apply a thermal gradient. And another control that we do is where we put the trap in horizontal position. Now, you might remember before that you need also convection for this accumulation mechanism to work. And the convection needs to go across the height of the pore. So if you put the trap horizontal, you get convection across the thickness of the pore, and that will just disrupt your entire flow, and it will not lead to accumulation. So that's kind of a control that even if you have a temperature gradient, you will still not produce or not accumulate anything. And you can see this here, for example, also in the DNA, in this DNA gel here, where when you don't apply a temperature gradient, or if you put it in horizontal, your DNA will be the same throughout all positions. But if you apply a temperature gradient, your DNA will be accumulated at the bottom, and it will just go from there to the top. It won't be any DNA in the top. And DNA accumulates actually very good in those kind of systems because it's such a big molecule. Now, you can apply the same thing with ions. So, for example, those are some of the most important ions in this reaction. Magnesium, of course, most biochemical reactions need magnesium, potassium as well. And phosphate is another important thing, not important in the sense that it's a waste molecule. So it's that kind of the waste that comes after a polymerization reaction, and it chelates the magnesium and it interacts with other parts of the reaction and will kind of block it. So you generally don't want this phosphate. But you can see that even that accumulates to some degree. Now, if you check also proteins also accumulate as you might have expected. But to different degrees depending on the protein, and that depends on multiple things, the size of the protein, how folded it is, the charge of the protein. However, all of them have to accumulate at least 3 times in order for the reaction to work. So even if you have different amounts of accumulation, most of them will accumulate enough for the reaction to work, which is quite nice for us. Now, the next part was to see if even if we have accumulation, this until now it was done in a closed system. So we just filled up the trap and we let it accumulate, but there was no input or output of material. So we wanted to see if we can introduce some form of flow in order to later maybe input some nutrients to the reaction and keep it running. Or even to show that it's confined against this kind of flow. So as you can see here in this diagram, we built kind of our setup a bit differently so that we can inject a solution on the top of the chamber, either when we have temperature gradient on or not. And you can see that we let, for example, a transcription translation reaction to run, and then we turn on the flow, and you can see that based on the flow speed, you can have different depletion speeds inside of the trap. We settled for the 11 microliters because we wanted to see if we can keep it stable even if you have a flow. And in order to test this more confidently, we also ran some simulations which fit pretty well to what we observe. So we're kind of certain that we can simulate this over longer periods of time. And you see here in this plot, the solid line are the experimental data and the dotted lines are simulation data. And after you start the reaction, after you finish the reaction and you start the flow, if you don't apply a temperature gradient, you will see that the reaction slowly depletes, and after around 6 months of simulated time, you will completely deplete from the chamber. However, if you have a temperature gradient on, and you will actually actively accumulate your molecules, it will take much, much, much longer for the trap to be depleted. And even after 6 months, you will still have around 80% of at least your superfolder GFP in the trap. But since everything accumulates, everything else should also be still accumulated enough for the reaction to run. And you can see here as a comparison, this is phosphate that I showed before. And as I mentioned, we don't want phosphate, so we want it to be removed. And it was actually quite interesting to find that if we use the chemical properties of phosphate, it will deplete much faster than the rest of the reaction, which gave us an idea that I'm going to talk about later. The next thing what we wanted to see if we can use this flow of water to feed nutrients in the solution. And for this, we took a pure that was depleted of amino acids and tRNAs, and we wanted to introduce them later through this flow. And you can see here in the dotted line, this is where we didn't apply a temperature gradient. You can see that when in the bottom position, in the top position, there is a short increase in intensity, but afterwards it depletes as more and more material is getting flushed out. However, as you see in the red lines, when you apply a temperature gradient, you have a brief increase at the top at the beginning, but then most of the material will accumulate at the bottom of the thread. It will kind of be protected from this depletion, and it will also accumulate together with the nutrients. So you can use the flow to feed the reaction for a longer time. Of course, the reaction stops at some point because there's other processes going on that are not dependent on the nutrients. But in theory, you could use this to feed the reaction over longer periods of time. Now, the last thing that I'm going to talk about is this, as I mentioned, we don't want waste. So we want to see if the waste can be removed while nutrients are kept. So for this, we ran a simulation where we started from the condition of a reaction that has already finished. So it has only phosphate and virtually no ATP, which is in our case kind of our nutrient. And if we start flowing ATP inside, you see that at some point we have a situation where we deplete the phosphate and we keep the ATP inside of the trap until it reaches kind of a steady state where as much phosphate is getting produced as it gets taken out of the trap, and as much ATP is getting used up as you get inside from the flow, which was actually very nice to see that it is possible to do within the same temperature gradient and in the same kind of pore. Was very interesting for us. Now, next thing what I'm going to talk about, this was thermogravitational trapping. Now, the other mechanism is, as I mentioned before, air-water interface, and this is a bit different than thermogravitational trapping. And for this, we also modified the chamber a bit. You can see here how this would look like. We have kind of this hanging droplet here, which is our air-water interface in this air chamber, and the solution is on the top here, and the air is on the bottom. And you have to imagine this mechanism kind of like, it's called a coffee ring effect. So the coffee ring effect, maybe you've noticed that if you drop kind of a bit of coffee on the table, and as it dries up, you see that it kind of makes a ring of like more dark material at the edges. And this is because as the droplet dries up, as it is pinned to the surface, it will dry up over the whole surface area of the droplet, but it will need to compensate the material lost at the edges in order to stay pinned. And this creates kind of a capillary flow that pushes your molecules to the edges, and as it completely dries up, your molecules will be accumulated in a ring. And this is kind of exactly the flow that we try to use in our trap. We use kind of a continuous coffee ring effect in the sense that we still evaporate our water at the air-water interface, but we also immediately condense it on the cold side, and this creates a net flow of water from the cold side to the hot side. And this flow of water at the surface of the bubble will also pick up the molecules and kind of accumulate them at the hot side at the air-water interface. And you can see this here in this kind of teaser image. You have this very highly concentrated localized superpolar GFP band here at the air-water interface. And the neat thing about this mechanism is that you don't have one accumulation place per pore. You have one accumulation place per air-water interface. So you can see here that even this small air bubble here, which is in contact with the hot side, accumulates material and is kind of another reaction chamber in the same, pore that we can also notice superpolar GFP production in. And first, as we did before, we tried it with a pure that was undiluted. And you can see that even undiluted pure, which produces superpolar GFP over the entire length of the trap, will be more accumulated at the bottom here, and it will be quite highly accumulated to the point where the superpolar GFP will just diffuse away from the air-water interface and will kind of forms this nice, little hotspot here in the middle. But you can clearly see that the accumulation is mostly at the bottom here at the air-water interface. And this just looks, this seems that it's just 6 orders of 6 times difference between the top and the bottom, but in actuality, it's much more. It's just that we've kind of reached the limit of the sensor here at the bottom. You will see a bit later in diluted pures that you have even higher accumulation. So you can see that if you plot the entire length of the trap here, you have your intensity peak increasing exponentially as you get closer and closer to the interface. And this is governed kind of by this simple exponential equation here, where C is your concentration, of course, and C0 is kind of the concentration at the most that you're measuring from, so the most distant part. D is the distance until your measured concentration. Velocity is the velocity of the flow at the air-water interface, and dc is of course the diffusion coefficient of the species that you're trying to that you're accumulating. And this fits quite nicely. It's not shown in this plot, but it fits quite nicely to what we observe. And now, I mean, as I teased before, we also tried diluted pure in this setting. And we can even dilute it more than with the other mechanisms. So for example, at 3 times diluted pure, you see it takes some time for the air-water interface to stabilize, but afterwards you see that it's very highly localized superfollow GFP production here. And also at the other bubbles that appear in this chamber, and that is where superfollow GFP is getting produced because of the pure that accumulates. And you can even dilute it more, 5 times diluted here, and you see the same kind of behavior. Your entire molecules, all the molecules of the pure reaction will accumulate at the air-water interface, and they will start producing superfollow GFP. And you can see that it's really the air-water interface and the temperature gradient that are doing this, because if you stop the temperature gradient, as I just did here in this experiment, all the molecules will just diffuse away from the interface. And you can see this here evidence here by superfolder GFP that just diffuses away and just equilibrates inside of the chamber. And also here, as we did before, if you don't add a template, for example, you will not get any production. But also if you put no temperature gradient across your chamber, you will also not get any superfolder GFP production, of course, because your molecules will not accumulate. So we have quite a good understanding of this mechanism, and we're quite certain that it's only diffusion that is trying to not works against the accumulation, because we also can run simulations, and we see that if we turn off the temperature gradient at this accumulated state, it fits quite well to what we observe from the diffusion data. We also see it in different molecules. For example, when we do protein gels, we see that the proteins are mostly at the bottom. And as the more we dilute the pure, the more and more you see this discrepancy here. For example, in the 1-time diluted pure, of course, we still see some superfollow GFP at the middle and top positions. But as you dilute more and more and more and more, you have less and less superfollow GFP in the rest of the chamber. Of course, you'll have some, because superfollow GFP will just diffuse away from the interface to some degree, so you'll still find a bit of superfollow GFP in the top and bottom and middle positions. However, you see that the discrepancy is getting quite high the more you dilute the reaction. Now, I just want to summarize this part. So we can accumulate transcription translation reactions to the point that they become active if we dilute them in the beginning, and this can be done only by putting a temperature difference across this air-water interface. If we switch it off, of course, everything will just diffuse away. Now, I mentioned this diffusing a lot because this is a state that we don't really want on the earlier. So if you accumulate your material, of course, you want to keep it accumulated, independent of how your environment is. If something happens to your like the heat, your geothermal activity, and the heat somehow subsides, and you will just lose all the material that you've accumulated. So cells have found a pretty smart way to keep their stuff inside, and that is by using phospholipid membranes. And this is not, of course, something that we want to try to replicate in our thermal pores. And for this, we kind of changed the chamber a bit in order to focus more on this air bubbles that I have mentioned before. And that is mostly in order to increase, the amount of reaction places or accumulation places that we can have in one chamber. And this is just how we devised the trap. We get this air bubbles here labeled with 1 at the beginning of the experiment, but we also have, as in other experiments that you saw before, we have this auxiliary air bubbles that just appear through degassing and during the times of the experiment, and those will also accumulate material. And we do kind of a bit of cheating. We use DOPC in order to for our vesicles, which is kind of the standard lipid that people use more or less when doing GUVs and when doing synthetic cells as well. And we just wanted to see if we can form anything meaningful with this mechanism. The first thing that we tried was with just DOPC and water and with a lipid dye to see kind of what will happen. And we were very surprised to see that we can form a large variety of vesicles in this chamber, some very big ones, some very small ones, which we can extract and then look at in a confocal, as you can see here on the right side. But then, we were interested in the ones that were unilamellar because, of course, life kind of focuses more on unilamellar vesicles. Of course, some bacteria have multiple membranes, but the easiest way is to have just one membrane that separates the interior from the exterior. And we did this using the typical alpha hemolysin assay, where you produce your vesicles, you introduce a dye on the outside of them, as you can see here, and then you introduce your alpha hemolysin and wait a bit. And that should only work if you only have one membrane. The alpha hemolysin will create pores, which will allow the dye to influx in the vesicles. This should not be possible if you have multiple membranes, of course, because the alpha hemolysin will only make pores in the outermost membrane. So you can see here that we saw influx of material from the outside to the inside, which was signaling us that we do have unilamellar vesicles forming in this setting. Now, making vesicles is nice, but we also want to put something inside, and preferably we want something to be accumulated first and then we put inside. So what we started to do this with is with DNA, which was labeled, as you can see here. And if you focus here on this video, you will see that the DNA slowly gets accumulated here. There's no lipid dye in this experiment, so you will only see the signal from the DNA. And as you can see that if you accumulate it and then you turn off the gradient, and it would normally lead to the DNA just diffusing away. However, in this case, it doesn't happen. The DNA will kind of get entrapped in these vesicles and at concentrations which it was accumulated at. And you can see that if you can also do multiple cycles of this, as you can see in the video on the right here. And it's a bit of a sped up version. But you can see that all the air bubbles here will accumulate material and will produce vesicles, and you can repeat the cycle again and again and again. And you produce vesicles, some of which are highly concentrated, some less, but a lot of them have very high concentrations of DNA inside. And of course, the vesicles, once they're formed, normally they would be kind of closed off from their environments. But in our case, they can also get back to the air bubbles, and they can get reincorporated and reaccumulated, which would allow mixing of their contents and then reform into vesicles, and then they can go off and colonize another air bubble. And the way this kind of works, as you can see here in this diagram, is very similar to what I showed you before. Of course, this being an air bubble, it kind of happens all over like the 3D geometry of the bubble on the hot side. So you'll kind of have this ring here of accumulated material, which will form your vesicles. And this is kind of a close-up a bit of what you saw before, a bit less sped up, and you can really see the vesicles kind of bubbling out from the air-water interface at the hot side. So this is focused on the hot side where you have your DNA accumulated. And you can really see here that the vesicles are really getting formed, and they kind of, elongate and then kind of form these nice spherical structures. And again, if you turn off the gradient, they will not diffuse away as they would happen if there were no lipids inside, but they will kind of stay inside of this vesicle form, and they will keep the DNA inside. And of course, when you accumulate it again, they will kind of just go back to the air-water interface and then reform and then remix. And then, of course, as we did before, we can also take this solution and look at it in the confocal, and we see really nice and concentrated DNA inside of those vesicles. And we also see, different structures also. We also noticed some where we had, an outer vesicle which had some concentration of DNA inside, and a smaller vesicle which has an even kind of higher concentration of DNA inside, which is a very, very interesting setting where you can have like, different concentrations of DNA separated, in kind of this nested structure. So this we did in water, and in water we kind of can reach, fold concentrations of kind of 40-fold, even more if you check out the individual vesicles. The average of this was around 30-fold, which is already quite high, with vesicles diameters of around 14 micrometers, which is kind of the standard, small to biggish GOV sizes that you can find. Now, of course, in water, vesicles form much more easily, but we also want to put reactions inside at some point. And to do this, we have to work in a buffer that has magnesium. And magnesium is very well known to not really, work well with vesicles because it makes them aggregate a lot. But we found out that if we put some 1% pegylated lipids in our mixture, it kind of helps with the magnesium, and it lets us form vesicles at the interface, but also in the bulk of the reaction, as you can see here in the background. This is a bit out of focus, but there are better videos that are coming. And if you extract this, you'll see that you still produce vesicles, but it's much more dirty because, of course, you have some magnesium and it will lead to aggregation. We also found out that if you add some form of osmolyte, for example, fructose or ribose or RNTPs, this will actually help with the vesicle formation and also with against aggregation. And you can see here it looks different than in the water experiments, but we do see a lot of the dynamic bubbling of the vesicles at the interface, which we can also see then afterwards in the confocal microscope. And this is, for example, with ribose, and this was a very nice one where you can see here that as the bubble touches the hot side, it will kind of start accumulating material and will start forming the vesicles containing DNA. And we are also very happy to find out that RNTPs also help with this. And RNTPs are nice because, of course, they're substrates for transcription translation reactions, but also for transcription reactions. So if you put them also in a bit more higher concentration, they do tend to help with the vesicle formation. And you can see here that the vesicle sizes when you put like those different osmolytes kind of stay the same. You have empty vesicles and filled vesicles are the vesicles that have a higher concentration than the bulk, and the empty vesicles are those that have a lower concentration than the bulk. And you can see that you have a difference in the average number of vesicles that you can get from each of the osmolytes. But not only that, there's also a difference in the ratio of filled to empty vesicles, with the RNTP. The RNTP is actually helping with forming a lot of the filled vesicles where you have concentrated DNA inside. Here we don't reach as high fold concentrations inside of the vesicles as you do in the water experiment, but still we reach between almost 6 and 15, which is still quite high for accumulation. Now, here I just want to highlight a bit the mechanism. So there are two main ways which we think vesicles form. One of them is through this diffusion. If the gradient subsides, you kind of have diffusion of your molecules, and while it diffuses, it kind of forms the vesicles. But you also have this vesicle formation mechanisms here, which happen during the accumulation, which we think are more interesting because the mechanism, we are not quite sure how it works. But of course, it's more interesting because you don't need to subside your temperature gradient in order to form the vesicles. You can just keep the gradient on, and the vesicles will just bubble out and they will form. And the end goal of the project and of this mechanism will be to put some form of reaction inside, as we did before we accumulated it. We were nice to accumulate it, activate it, and put it into vesicles. And we kind of managed to do this already with IVT, which is much simpler than transcription translation reactions, because we can control all the components. And you can see here that you accumulate it, and this, what you see here is the fluorescence from broccoli. And when you turn off the gradient, everything will kind of go into vesicles. And then you'll have, your broccoli and your reaction inside of those vesicles. And you can restart the gradient later, and it will just float around and colonize other bubbles. But then, the holy grail of it is to put the transcription translation reaction inside, and that's what we're working on now. That would be the end goal of this project. So in order to summarize the talk, we can accumulate DNA or other molecules in vesicles, and you can put them inside of those vesicles at quite high concentrations. We see different types of vesicle populations. We have, filled ones where you have the concentration inside higher than the bulk and the empty ones, but you also have different mechanisms of how those vesicles form. And you can, have repeated cycles of this temperature gradient on and off at the interface, which will just produce vesicles, release them, and they will come back at the interface and kind of open up and remix and then reform. And this, we kind of try to say that it's more of an effect, kind of like a day-night cycle, where during the night, of course, the temperatures outside will be colder, so your water will be colder, you'll have a stronger gradient. And during the day, when the water will get heated up, you will have less of a difference between, the geothermal activity and the water, and it will kind of make a weaker gradient, which will lead to some of the vesicles kind of diffusing or like going away from the interface. And with this, I would like to hand it back to Dieter, just to make kind of the finishing of the talk.
Okay. Hi, everyone. So where we put this on the origin of life is we look for these pores, and we've had other projects where these air-water interfaces are attracting nucleotides, oligonucleotides, RNA, and we see strong accumulation of those too, and actually evolution. So we have PCR reactions running there, making actually longer and longer DNA, showing that the replication is actually driving an interesting Darwinian evolution, of course, with proteins. So we'll hopefully have that also working for the 2,3' templated ligation. And as you've seen, those are also the environments where complex pure system can accumulate, do their thing, and actually be wrapped up in vesicles. So just a bit of shout out, that's basically, I think, the combination, what we are after, both the accumulation of concentration, and if they complexify, get more complex, they will accumulate better. Same way that RNA sequences, if they replicate, get longer, they also accumulate better, and the whole thing is operating on these accumulated interfaces. And that's the scenario to think about, I think, of complexification out of nothing, out of the early Earth. Shout out some advertisement. If you're around Munich in June 11th, 12th, we have the 10th iteration of our Molecular Origins Munich MOM conference. These are beautiful posters. Registration deadlines is basically not existent. You can just show up, there are no fees, and bring your poster. Poster registration needs a little bit feedback from our side, but we'll keep it as easy as possible. So hopefully to see you there, and that's kind of based on the network, the Origin of Life. It's a collaborative research center funded by the DFG, Molecular Evolution Prebiotic Environments, which I'm organizing, where 20 PIs from very different disciplines come together. That's our website, molecularevolution.de. Have a look at it, and if you're interested in the topic, always opportunities to collaborate with our many different expertises. And I want to end with a picture of the group and acknowledgements. Mostly important ones is to Hannes Mutschler, Kerstin Göpfrich, Tomoki Matsuura were contributors to the project. Diana Chakraborty was contributing, also Pratulya Shah was working on it. And thank you for your time, and happy that we take questions. I already had some of them answered as good as I could.
Thank you very much.
Okay. Thank you. Thank you, guys. This was really, I appreciate the science, but I also really appreciate the visual presentation of it. There's some more questions in chat. Could you read the question out loud before answering?
I'll just read it out loud and Alex will answer. Is that okay?
Yes.
Great talk. Do you see some degradation of components like ATP hydrolysis and tRNA degradation?
Well, we didn't really follow that, but I think from other experiments that we did at air-water interface, there was some degradation, but not that much, right?
I mean, one should keep in mind that we are still within the temperature range of pure. So the setting is made with the initial screen, what are temperatures where pure is operating, and then we actually take that as a temperature bracket for us. So there's no reason to think that the gradient of temperature would start degradation where a constant temperature would not.
Also, like the tRNAs, usually since it's a commercial pure, they already come in quite high concentration. So there will be probably some degradation, but I don't think that much that you will kind of just destroy your reaction. And the RNA that is produced from the DNA that gets produced all the time, basically if there are nutrients around. So that also you wouldn't kind of notice the degradation of that.
Do you account for evaporation in your microfluidic system? Is it a closed system?
So it is a closed system. At least the air part is closed. In theory, of course, if you leave it open, it will just dry up. But in a closed system, as it was in the air bubble, the water that evaporates on one side, so on the hot side, will actually condense in the cold side. Of course, when it reaches, the maximum amount of humidity that you can in that air, it will condense on the cold side, and you kind of have this cycle. That's why we try to focus more in the next part. We will try to focus more on the air bubbles because they are already enclosed, air pockets without us needing to, close the chamber and have, screws and everything. So you don't see, evaporation and loss of water in those air bubbles.
And that's the side of the gas. I mean, the water side, which is basically always pulling in nutrients for your interface, can be coupled to flow, as we showed. So that side can be actually open. We're also experimenting a lot with really evaporative system, even systems without temperature gradients. We run a PCR reaction, those systems 2 years ago. So we think that has a lot of potential to even go for very, very flat and shallow temperature gradients. In your vesicle formation experiments, do you see encapsulation of all solutes, or is it anything excluded from the inner lumen? That question came early in the talk.
Yes, that we still have to see, at least for the reaction. Of course, the DNA is getting encapsulated. I mean, it's the fluorescent signal that we see. And also, if we have, different lengths of DNA, we see encapsulation of both of them. However, it depends a bit on the length of the DNA. So I guess the accumulation and the encapsulation is dependent on the diffusion coefficients of the materials. It remains to be seen if we extract the lipids of the vesicles, open them up, and then just check what was encapsulated inside.
I mean that was actually initially when we started this experimentation quite a long time ago. We didn't expect that it would work, right? Because you would expect that the DNA accumulate much more, the others don't, and then you immediately make a misbalance of all the compounds. Now, it seems to be, interestingly enough, that these crowded, higher concentrated compounds kind of, in a way, accumulate cooperatively, right? If you look at the protein gels, they all accumulate very similarly, even down to dilution factors of 5 and so on. So that's kind of the funny thing, which the system is better than expected in a way. So some things are really strongly accumulated, like DNA.
Yes.
But that's good for the system.
Yes.
Keeping DNA at your location, no matter what, is an evolutionary good trait.
And some things are less accumulated, like magnesium, at least in the air-water interface experiments, magnesium or ions generally are less accumulated because they have a quite high diffusion coefficient, and they will just, they just diffuse right away once they're accumulated.
And that gives you an interesting, exchange to the environment because it's a steady state. So you have enhanced concentration. Things come, but things can also go. And
Yeah,
you know,
yeah. A lot of molecules just pass kind of through this accumulated phase, kind of like a reactor, and they get pulled in, they react, and then just get diffused away. It's not always the same molecules that are accumulated.
So it's an open system.
Yeah, it's a steady state,
which can make the closed system. So you have, we would argue with both good things of both worlds, as you want to have it, right? Because making a cell which you close up and then you have to fiddle around a lot to get those transporters, all the things, you know, things are still happening next door at the interface, and then eventually you can jump and make a full cell. Anyway, you get away. Fascinating talk. Is 10 degrees C over 100 micrometer of temperature gradient realistic in aquatic environments? It seems extremely steep. It is steep, if I may answer. And that's a bit on purpose because the steeper systems are the fastest accumulating. So we see, even if we switch off the heating, where we have very shallow temperature gradients, still accumulation. And please note that these air-water interfaces already need less steep ones. Also, these thermal phoretic accumulations. We have a paper out already time ago, where even if you have 100 degrees of 1 meter, gives you the same accumulation if you make a pore along those temperature differences, kind of a large pore space, but it would equilibrate in a half a year, which will be very tough for the experimentation.
Yeah, I guess it's not good for a PhD student if the experiment takes half a year to make.
And evolution will pick the fastest system.
Yeah.
So there are some arguments.
Yeah. But as Dieter mentioned, the air-water interface accumulation is much more easy to get to, because, for example, I didn't mention it in the talk, but the one that I showed, so the pore size was 0.5 millimeter and the gradient was 6 degrees, and you already get almost the same, you know, localized accumulation as in the other chamber. So I would say the air-water interface is a bit more forgiving in that sense.
Yeah.
Mm.
Some non-equilibrium is necessary, but
yeah,
I agree. Yeah. Would it technically be possible to add cold area with frozen phase like eutectic reactions? We don't say anything,
Not
should we?
yeah.
Yeah, interesting things in that regime. Yeah. Watch this space.
Yeah, we're yeah.
now we get
a bit secretive.
Well, if you freeze water, as you know, you get a lot of air bubbles, and you actually see even these freezing experiments accumulation around it. So it's quite interesting if you think about that.
It would be interesting if you can combine, because there's a lot of condensation reactions you could get in eutectic, and then you shoot it out into your reaction supporting circulating environment. So that would be really cool.
And that's what you see. You know, you can make the same chambers, cool it down, and then you see very interesting things. You see the eutectic phase, you see the air bubbles, you still have a temperature difference. All of that is open. And origin of life, early sun was very weak. Thinking about ice is not impossible, although early Earth was hot, but it might have cooled down quite fast to temperatures. You know, the good thing, I think, is that, I mean, at least for the RNA reaction, we, of course, focus on, we seem to be operational on a regime from -30 up to +80 degrees to get polymerization, for example. So with pure, that might be difficult. With
only if we get some pure from thermophilic bacteria or something.
But I mean, please, if you're interested, approach us. We have mobile setups making temperature gradients, or visit us to collaborate. We are happy to feed your system with some non-equilibrium, be it evaporation without temperature gradient or temperature gradient.
All right. All right, I think you went through all the questions. I just want to say thank you so much again for a fantastic talk.
Thanks for hosting.
And thank you for doing fantastic work.
Thank you.
He's still a PhD student. Look.
That's not long.
For not long, you say? Does your PI knows about it?
Yeah.
We all know all the time, do we? Yeah. Okay.
All
Thank
right.
you.
Thank you so much, guys, and have a
Bye-bye.
great rest of your day.
Yeah.
Thank you.
Bye.
Bye-bye.
Bye-bye.